Thank you for the kind introduction. I will not be bothering much more of our time right now. I know the value of the time right now, and I believe this is a very important topic that has been carefully selected by Dr. Somani and the whole scientific committee. I personally looked after this topic as a very important future potential biomarker for the predictor of the nivolumab efficacy, which is very important in our daily routine practice. So it was presented by the Yale group in the ESMO 2022. It was a very important discussion for us. The evaluation of the DNA damage response as a predictor for nivolumab efficacy. So the study was very simple, that it was a group of pathologists who looked after the DNA mismatch repair genes, which were, can I request the team to please share the slides on my behalf? And move to the third slide. Yes, so perfectly fine. So what they did was they looked at the peripheral blood mononuclear cells initially, which had a DDR related parameters and a higher levels of oxidative stress, a known endogenous DNA damage matters as compared to the healthy individuals. And they found, put it up the hypothesis that those with the DDR signals may predict therapeutic benefit ratios in the head and neck squamous cell carcinomas. Next slide, please. So the methods was simple: the oxidative stress, the DDR associated parameters, which includes the endogenous DNA damage and the basal DNA repair genes known for the nuclear excision repair and the double strand break repairs, were evaluated, and they were treated in a phase 2 concurrently going trials for the nivolumab, and there was 26 healthy controls. And the peripheral blood mononuclear cells were evaluated at baseline, 4 weeks of nivolumab, and at progression. Next slide, please. So the method included the endogenous DNA damage, which includes the CNA, single and double stranded breaks, the alkaline comet assays. The double stranded breaks were assessed by the confocal microscopy. Oxidative stress was assessed by, again, the glutathione ratios, the abasic sites, that includes the AP sites, and early stage DNA lesions, which is as a result of the oxidative stress, was detected by a DNA quantification kit, and a nuclear excision read, which was an in vivo ex vivo treatment with the cisplatin by a southern blot analysis, a double strand break by a confocal microscopy, and apoptosis rate by a apoptosis kit. So next slide, please. The peripheral blood mononuclear cells from the head and neck squamous cell carcinoma showed a baseline high levels of endogenous DNA damage, which includes the single stranded breaks and the double stranded breaks, compared with the healthy controls. After 4 weeks of the treatment with nivolumab, it did not affect the levels of the DNA damage. The peripheral blood from the head and neck squamous cell carcinomas at baseline exhibited a higher level of oxidative stress, as you could see from the corresponding graphs, and basic sites as compared with the healthy controls. The DDR associated pathways, next slide please, at the baselines, also following the ex vivo treatment with the cisplatin, peripheral blood mononuclear cells showed a reduced NER and increased double stranded break repair capacity. The higher cisplatin doses are required to trigger the apoptosis in peripheral blood mononuclear cells as compared to the healthy controls, indicating that the patients are characterized by a lower apoptosis rate. And if we look at the single strand breaks and the double stranded breaks and the response to nivolumab, the lower levels of the DNA damage at baselines were higher likelihood of response to the nivolumab, improving the clinical benefit rate and longer overall survival, as you could see from those graphs. Also, the lower levels of the DNA damage at the baseline were associated with higher likelihood of response to nivolumab, improved clinical benefit, and longer OS. Next slide, please. The lower levels of oxidative stress at baseline were associated with higher likelihood of response, improved clinical benefit rate, and prolonged overall survival. If we look at the nuclear excision repair capacity, the individual reduced nuclear excision repair capacity, that is increased cisplatin-induced monoadducts at baseline, is associated with the higher likelihood for response and improved clinical benefit rate and higher overall survival. And if we look at the double stranded break repair capacity, the reduced double stranded repair capacity is associated with a better clinical response, improved clinical benefit rate, and a higher OS. Also, the higher cisplatin dose are required to trigger the apoptosis in peripheral blood mononuclear cells from the responders compared to non-responders, indicating that the responders to nivolumab are characterized by a lower apoptosis rate. So a lot to take out from it, that yes, these are newer potential biomarkers for the nivolumab to be given in a head neck squamous cell carcinomas. Some of the valid promising hypothesis generating and definitely clinical trial biomarkers are the reduced endogenous DNA damage, the oxidative stress, the DNA repair capacity, and apoptosis rates in the peripheral blood mononuclear cells of patients with head neck squamous cell carcinomas with a better response to nivolumab therapy. Definitely a larger phase 3 trial with a broader cohort and a prospective way of doing it will be a much better way of doing it. That concludes my presentation, and over to the organizers for a lovely crafted agenda and a beautiful conference so far.
Thank you, Dr. Vikas. Although you have said everything is very tempting and very interesting, but I think the research area of the evaluation of response rate in India is not, I think it's not available. So we have to go far ahead to develop all this facility which you are talking in India, and particularly validated also. So otherwise, it's a very good idea what you are saying.